My name is Astha Symes, and welcome to Live Longer World. Dr. Ewald, welcome to Live Longer World.
Glad to be here.
So, if someone asked you how you would describe your book, Plague Time, and the research that you describe in it, which is the infectious causes of chronic diseases, how would you describe it to someone?
I describe it as a start towards understanding more fully the causes of chronic diseases. But also, I'd say that it's recognizing what's missing from particular approaches. And so I'd also add that when we're looking at chronic diseases, especially, we don't understand them very well because causation is so complicated. Generally, if you look at the last 200 years, medical sciences have solved the easy problems first, really easy problems, and then more difficult problems take longer and longer to solve. So we're left with difficult problems now in terms of understanding the causes of disease. And so plague time is really trying to emphasize that one big category of causation that had been largely kind of neglected in terms of mainstream health sciences was the role of infection. And so that was kind of focusing on the role of infection, but it really is sort of a starting point because the role of infection can only be understood in the context of the interaction between infectious agents and other causes. So we have to be always thinking about, what are the roles of genetic vulnerabilities to various factors? What are the roles of environmental circumstances? And so if you're thinking about infection, really the next step is to understand not just the collection of risk factors that play a role in these chronic diseases, cardiovascular diseases. You understand one thing, and then that leads you to recognize that that one thing is related to another thing, and that other thing is another, and it sort of feeds back on the first thing. So it gets to be very complicated. And I think that's the reason why almost all of these chronic diseases just aren't that well understood. People claim they understand them well, but all you have to do is sort of say, well, wait a minute, this explanation doesn't explain some aspect. And so I think recognizing that there are these paradoxes or holes in the explanations is kind of a first step. And then that should generate curiosity to say, well, what then is an explanation that allows us to understand this gap of knowledge?
So, what you're saying is that there is an overlooked explanation of the role of certain viruses in causing diseases like cancer, Alzheimer's, atherosclerosis, and the current explanations have certain holes in them as well.
Yes.
There are two questions that come from this. One, maybe let's talk about the holes with some of the current explanations. And most of the current explanations are it's either genetic, it's lifestyle, it's metabolic health that contributes to cancer and heart disease.
Yeah.
No one thinks of viruses.
Yeah.
So,
Well, there are people that do, but they're
right.
virologists. Okay.
Well, people like you, and I think more people
That's right.
are starting to learn.
Yeah. And so it's very frustrating for people who are working in that area to have really interesting information that seems very powerful, and yet to have other people that are kind of proceeding along the sort of standard explanations just dismiss it without really looking both at the drawbacks of the explanations that they're proposing, but also how those drawbacks could be resolved by linking what they're studying with infectious causation, for example. And it isn't just viruses, it's bacteria and protozoa and even wormy parasites that can be playing a role. But, we could sort of take a number of these different chronic diseases. A good example is, I think, Alzheimer's disease, and in particular, the non-familial sporadic Alzheimer's, which is the most common form of Alzheimer's. And the focus has been on the occurrence of plaques and tangles in the brain. And the idea is the plaques and tangles are there in all Alzheimer's patients, so they must be causing the Alzheimer's without thinking, well, maybe they could be a consequence. And also without sort of forgetting that we define Alzheimer's as a dementia that has plaques and tangles, right? So there's a circularity there. But, when you look at it closely, it really looks like for example, the plaques, the amyloid plaques, are involved in some pretty complicated interactions. The first question is, why would the body have amyloid plaques just forming and causing the brain to malfunction, right? And one of the arguments is, well, that's a disease of old age, and we're not very well adapted to have things work in old age, old decades of old age. So that's one explanation. That there are holes in that explanation. For one, is that this occurs even in younger ages, where we expect natural selection to have sort of solved the problem. But then, as data become more apparent, then you begin to see that there are problems with that explanation. One problem is when you actually have interventions that target the plaques, it looks like they have, at least some of them have some positive effects, but most of them don't. And so the question is, why is it, with all this effort and dozens of studies that target this, only a few seem to provide a benefit, and the benefits are really quite minor? So that's an issue that has to be addressed. Part of the resolution is these amyloid plaques are formed as a result of the accumulation of the beta amyloid fragments of bigger protein. And those fragments, although they do have some toxic effects, which probably contribute to Alzheimer's, they're also antimicrobial. And so when you actually look at all of the data, what emerges is that these microbes, in this case, bacteria largely, but some viruses, look like they're very strongly associated with Alzheimer's. They're causing damage that is correlated with the genetic associations with Alzheimer's, and they're triggering amyloid beta protein formation. And the way to think of that is the brain may detect a pathogen in it, a virus or a bacteria, and it starts producing beta amyloid as an antimicrobial. And so there can be a battle that's on, and the amyloid beta is produced, and it ends up forming plaques. And those plaques can cause damage, but a lot of the damage may be the damage associated with this battle that's going on between the pathogens that have gotten in the brain and the beta amyloid. And so, one of these pathogens, actually two of the pathogens that really look like they're commonly found in Alzheimer's and may be causal, are pathogens that are causing gum disease and periodontitis. And so that's sort of an odd thing. Why would that be the case? But these pathogens tend to be very invasive. They get into the bloodstream, and they actually are causing damage in a lot of places in the body, but one place seems to be in the brain. So then you look at these associations, and you do studies with mice, and you can show that these pathogens can be generating molecules that are sort of damaging the brain, but then also the production of the amyloid is helping to control those damaging chemicals. So you begin to see in a mechanistic way how this can actually work together. So you sort of put it all together and say, well, the arguments about Alzheimer's just being the body falling apart aren't very compelling. And when you look at the genetic associations, yeah, they look like they're contributing, but they're not actually explaining all of Alzheimer's or even most of Alzheimer's. And so then you ask, well, what are the genetic vulnerabilities? One of them is this epsilon 4 protein. And the epsilon 4 protein looks like it's interacting with some of these pathogens that are showing up in Alzheimer's. So the pathogens may be more able to cause damage, or we're more vulnerable to the pathogens if we happen to have this genetic basis. Okay, and so there are all of these kinds of factors. And if you look at the interaction between these organisms that are causing gingivitis and periodontitis, then you also have social factors. And one important aspect of this is how well are people take care of their teeth, or especially are they flossing? And there's information suggesting it's correlated, but there should be experimental information coming out soon, indicating that if you floss, you really lower your risk of Alzheimer's. And that's pretty surprising, right? But it's actually very encouraging. If you floss, you also control your periodontal disease. And so if you're controlling your periodontal disease, it may mean that you're going to have less of that invasion in the body and less of the damage to the brain. And then the other question is, well, why are some people motivated to have better oral hygiene? Or why are they having better hygiene regardless of motivation? So some of that's education, some of it's access to good dental care. and some of it's diet too. So we've got all of these things interacting. And if you look at any one of them, it doesn't explain it. But if you look at how these things interact together, the genetics, the environment, including the social environment, and also microbes, then you begin to see a picture emerging, which really at least provides a coherent view. But then we have to say, well, is this coherent view really the right answer or not? Then we have to try to test those interactions. And that's hard to do, but we have to at least recognize that those are the hypotheses, sort of complex hypotheses that are consistent with the evidence that we see. So that's sort of where we are now. So as I look at the work that Holly and I have been doing over the last 2 decades, you can see that you have to kind of understand the expertise in a lot of different areas for a single disease. The same thing is true with cancer. The same thing is true with cardiovascular disease. It's even more complex with diabetes. So, and then these disease processes may interact with each other. Okay, so Alzheimer's is correlated with cardiovascular disease. It's correlated with diabetes. So we need to understand why there are those correlations there. And we can't just stop at some answer that really doesn't provide a thorough understanding. Like one of the answers, well, if you've got cardiovascular disease, your blood supply to the brain is messed up, so you're going to be more likely to have Alzheimer's, right? Well, that's a possibility, and it may have some validity, but we also want to look at the possibility that some of these other risk factors that look like they're playing a role in one disease are also playing the same role in another, or a similar role in another. So, for example, we look at these periodontal pathogens, and one of them is called Porphyromonas gingivalis. That's found in the brains of Alzheimer's patients. It's also found in the plaques in the arteries of patients with atherosclerosis. And so then the issue is, are we really seeing two separate effects of a given kind of infection with a given pathogen? And it's also playing a role in diabetes. So it invades the bloodstream. It alters the ability of the body to get insulin uptake by cells. And so it's increasing the damage from diabetes, and then the increased, the effects of insulin is what I meant to say, the glucose uptake by cells. And so then you can see that connection. But then the next connection is, well, why does diabetes mess everything up, right? It seems to be messing up almost all of these chronic diseases. It messes up cancer, messes up Alzheimer's, messes up, when it messes up, I mean exacerbates, right? And so then why is the question? And that's a mystery. People say, well, it just does it. It just messes the body up, and they try to get a particular explanation for a particular disease. And that's good to try to do that. But we need to also ask the question, are there some underlying commonalities that help us understand these connections? And so one underlying commonality is probably that in diabetes, you've got more glucose in the blood, and glucose sort of fosters the problems or exacerbates the problems that you see in these diseases. And part of that may be that it exacerbates the problems because these diseases are caused in part by infection. When you have higher glucose in blood, you've essentially got more fuel for the infections, right? So then instead of being just a big mystery that diabetes just seems to mess everything up, it's, diabetes is actually causing a change that actually encourages the disease process, an infectious disease process, or exacerbates the problem by making the infectious adversary more difficult for us to control. And then it also there's another layer, which is like with diabetes, a number of pathogens, in this case, bacteria, look like they actually manipulate the function of insulin. And that increases glucose availability, which helps them. And so pathogens may be starting out some of the problems, instigating some of the problems with diabetes, and then the problems with diabetes influence other things such as periodontal disease, and then the periodontal pathogens exacerbate the glucose uptake, and so the whole thing can become a vicious cycle. Right. And so then the question is, if you understand this, you might understand, how do you turn it back? How do you resolve it? And then that comes back to what I was saying earlier about, the practices, sort of the social environment. If people were more encouraged, more able to do flossing, then they might turn all of this around, right? Because they wouldn't have the organisms that are interfering with the insulin activity. So there would be lower glucose and available. So there would be less of the exacerbation of the oral pathogens, the pathogens causing gingivitis and periodontitis. And then the other diseases that they're playing a role in, like the Alzheimer's and chronic kidney disease and cardiovascular disease. Okay, so
That,
it seems like a mess, but it actually is. As you start looking at these connections, things begin to get resolved. It's just that there's so much work that actually making those connections is, is very time consuming and it's hard for people to do when their daily activities are focused on a particular slice of the problem.
right. I mean, this is just fascinating because you're totally right. I think biology often works in silos where you're working on your
Yes,
slice of the
right.
problem. And I think what you're saying is that, well, in order to see the full picture, the full puzzle of some of these diseases, we
Mhm.
cannot just isolate genetics or
That's
lifestyle.
right. Yeah.
And genetics and lifestyle play a role, but
Yes.
then there's this hidden role of viruses, which are
Yeah.
probably interacting with genetics and lifestyle. And
Yes.
one of the clearest examples, which I think more and more people do recognize, is the periodontal disease and Alzheimer's example that you
Yes.
give,
Mhm.
which is yes, oral bacteria seems to be a pretty strong factor in Alzheimer's disease.
That's right.
But then what's interesting is what you're studying is the role of this bacteria in actually causing or strongly associated with the disease, and also how it interacts with the other diseases. That's not
Yes.
something I thought of,
Yeah.
how it interacts with diabetes and atherosclerosis that you mentioned.
Yes. That's right.
One
That's
One
well
thing
said.
I want to go back to is you said that some of the current arguments, such as the genetic theories and explanations, have black holes. They don't explain the full picture.
Mhm.
And there is a quote that I found in your book, which I thought was pretty good, and maybe you can elaborate on it, which is that you say there's a crucial unrecognized problem with genetic causation of harmful diseases, whether they be cancers or other chronic diseases. How do the harmful genes maintain their representation over time? And you say that the harm caused by infectious agents is not constrained in this way. What does this mean?
Well, you can think of it as the infectious agents are continually changing. If the environment were constant over tens of thousands of years or hundreds of thousands of years, natural selection is really effective at getting rid of um, gene variants, alleles, that have harmful effects. Just because every generation, those are, because of their harmfulness, they're weeded out. But if you have infectious agents causing the problem, is you have a genetic vulnerability to an infectious agent. That infectious agent may thrive, and that may favor genetic resistance to the infectious agent. But then once you've got resistance, that puts pressure on the infectious agent to change. So it's no longer controlled by that genetic resistance. And so the infectious agents are always changing. They can change very rapidly because their generation times are very short. So with infectious agents, it's sort of like an environment that's always changing, and natural selection just has a hard time favoring resistance to those infectious agents because it's a kind of an arms race. But the time scale favors the infectious agents unless these long-lived organisms like us have characteristics that sort of allow us to combat them at the same time scale. And that's what the immune system does. The immune system has a lot of mechanisms that allow it to respond to changes in infectious agents within a lifetime, and certainly over a few lifetimes. And so, as a consequence, we have this any disease that has an infectious cause component being something that is likely to be with us perpetually because there's always this back and forth going on between, an offensive characteristic that the pathogen has, a defensive characteristic that we have, which causes the pathogen to have a different response. And so when we're thinking about genetic associations of disease, we have to ask the question, why would, if a person's claiming that a genetic variant is a cause of a disease, we have to ask, well, why would that cause still be present? And one of the answers is that genetic cause is a vulnerability to an infection. And we're just seeing how that is present in this current state of this ongoing, conflict between the pathogens and the hosts. And so it's when you're seeing genetic bases that are in genes that are involved in the immune system, that's kind of a red flag that you've got the genetic basis being a vulnerability to a given pathogen at the current moment, especially because those genetic bases are changing all the time. We know that because almost everybody's genetic basis for, controlling the particular proteins that are antimicrobial, they're different for everybody. It's extremely variable. These are the HLA alleles or MHC alleles, depending what terminology you want to use. And that variability tells us there's something going on. If you have a gene that has just a few variants, a few alleles in the human population, and they're pretty much present in large portions, as a consequence, then that tells us, it's not so much of a rapid fire arms race, but you still have to understand why you get that variation. And sometimes that still is associated with infections. And so the epsilon 4 allele that I talked about earlier seems to be exactly that. The epsilon 4 allele was sort of the standard allele for our species and even for ancestral species. But during the last 200,000 years, the epsilon 4 allele gradually started declining, and the other alleles, epsilon 3 especially, started increasing. So the question is, why is that happening? And if you could look at things like diet, it could be that diet's playing a role. But also, if you look at infections, you don't want to just look at one thing. You want to look at a lot of things. And when you look at infections, some of the candidates for causing these chronic diseases, such as one we haven't talked about, is chlamydia pneumoniae. Chlamydia pneumoniae uses the epsilon 4 protein to get into cells. So it's sort of like a doorknob that it doesn't use the epsilon 3. So if you're epsilon 4 positive, you have a protein that's really good for moving fats around in the body, fats and cholesterol, but it's coming with a liability that you're more vulnerable to the infections with pathogens like chlamydia pneumoniae. Also, herpes simplex one looks like it is a pathogen that is more able to cause damage in people who are epsilon 4 positive, who are carrying that epsilon 4 allele. And both of those are involved in Alzheimer's. Alzheimer's is more problematic in people who are epsilon 4, right? So that you have that connection. Same thing with in terms of chlamydia pneumoniae with atherosclerosis. It's been found in these plaques, and because epsilon 4 people are more vulnerable to chlamydia pneumoniae, we would expect that epsilon 4 people will get more damage. And so everything sort of fits together if you look at that genetic association in the context of this arms race that's going on between us and these pathogens. And, that arms race may be a real slow burn for, chlamydia pneumoniae, for example. It doesn't cause that much damage, and the damage tends to be in older ages. So the selection pressure may be very slight against it. Normally, it just causes some respiratory tract infection, but then may cause cardiovascular disease later in life or Alzheimer's later in life. And so it could be that the selection pressure against people who have epsilon 4 is really quite slight and may require many thousands of years in order to have a decline in epsilon 4. And inconsistent with that is the finding that it looks like chlamydia pneumonia has been around for many tens of thousands of years. If you look at isolated populations, they've had their own chlamydia pneumoniae variant. It looks like from the time they were isolated. So sometimes this goes back to 60,000 years. And so that tells us that this has probably been a slow burning, ongoing conflict. Epsilon 4 is probably better than the other epsilons for that gene. It's called the apolipoprotein E gene. But even though it's better inherently, it's maybe just a little bit worse if you've got chlamydia pneumonia in the population, right? And if you've got dense populations, it may be quite a bit worse. And interestingly, where you have had agriculture for long periods of time in denser populations, the epsilon 4 has declined to a lower level than when you have had agriculture not at all, let's say in hunter-gathering societies or only recently. So in those societies, some of those, you've got the epsilon 4 still being on par with epsilon 3. But if you look at places, like the Mediterranean or area or China, you've got epsilon 4 going down to being a fairly small minority of being represented as a small minority of the population. Okay.
That's interesting. I didn't realize it's declining. Is there any data that shows that the percent of people with epsilon 4 who get Alzheimer's, how many of them have one of these viruses?
Yes. Some of the early work on Alzheimer's and chlamydia pneumonia showed a very strong association between chlamydia pneumonia and Alzheimer's. And that same team then looked at the brains of the people who had died of Alzheimer's, and then they assessed the genetic makeup of those individuals. And the individuals they were homozygous for epsilon 4 had two copies of the epsilon 4 allele instead of just one. They had the most intense density of chlamydia pneumoniae. People who had one copy of epsilon 4 and one of epsilon 3 had less dense, and people who didn't have either had the lowest density. That addresses your question to some extent, but what you'd really want is to be able to look at the brains of everybody who's infected. These are people who died of Alzheimer's. So it could be that the people who didn't have epsilon 4 just had a number of other characteristics that led them to die of Alzheimer's. You can't tell whether the people who didn't have epsilon 4 were generally getting very light loads of chlamydia, so light that maybe they weren't even getting any symptoms of Alzheimer's, maybe a few plaques or tangles, something like that.
But then so then I guess if you take the whole population, what percent of people who do have Alzheimer's have chlamydia pneumonia?
That hasn't been documented. That'd be a very interesting thing to look at.
Yeah.
One of the problems with a lot of these pathogens that look like they're playing a role in chronic disease is that everybody gets them. So probably everybody has gotten chlamydia pneumonia. And a lot of people have fought it off. Some people have gotten it 4, 5, 10 times. Some people may be more vulnerable, because of their genetic makeup. And so it's a real problem. You can't just say who got chlamydia pneumonia and who got Alzheimer's, because you got to look at how intense the infections were. And, this is something that is also is sort of present across the board. So, for example, if we look at multiple sclerosis, these kinds of discussions, disagreements that we've been talking about because of the complexity of the situations have been going on in multiple sclerosis for about 3 decades with regard to a virus, Epstein-Barr virus, and its role in multiple sclerosis. So you can go back to the 1990s, you see people who said, Epstein-Barr virus is causing more intense infections in people who actually have multiple sclerosis than it does in people who don't. But still, if you have a certain threshold of a fairly intense infection, it's 100% in the multiple sclerosis patients and 90% in people who don't have it, right? So you have to be careful in trying to figure out, well, how are we going to assess whether Epstein-Barr virus is causing a bigger problem in terms of the intensity of infection in one versus another. So you have to have these kinds of thresholds. So for 30 years, this has been a source of disagreement. And I think it's pretty clear that in the last few years, finally a consensus is being reached. And the consensus resulted from looking at a lot of different factors that would be potentially discounting the possibility of Epstein-Barr or possibly supporting the idea of Epstein-Barr playing a role in multiple sclerosis. None of them by themselves prove it, but together they're leading, I think, to a consensus that yes, Epstein-Barr virus is a causal factor in multiple sclerosis. Okay, people could still argue maybe it's not 100%, maybe it's 50. We don't have the data on that. But some of the information that has led to this consensus is looking at the genetics. So there's a variant in one of these genes I talked about earlier, the HLA, a particular HLA gene, and that HLA gene codes for a protein that the Epstein-Barr virus uses as a receptor to get into cells. Okay, so you say, oh, here's the genetic component. Oh, the genetic component makes, we now have data molecular level saying that genetic component makes people more vulnerable to Epstein-Barr virus. And if you have virtually everybody with multiple sclerosis has a pretty substantial Epstein-Barr virus infection. So you put all that together, and then you sort of say, people are saying, yeah, it really is. Okay, so this is something that I would say even 5 years ago, still a lot of people were disagreeing with. 20 years ago, 25 years ago, people saying, come on, it's got to be playing a role. We wouldn't be getting this. And now we put all this together. And I think it's hard to say. You can look at review papers, you can look at medical texts, and you see how they're addressing it. With Epstein-Barr, it sort of goes in stages and multiple sclerosis. So some of the recent work is saying, well, the data are compelling, in the recent review papers, data are compelling for Epstein-Barr as a trigger of multiple sclerosis. So they're still relying on the trigger, right? And so that's part of the problem, is that the immunologists have come at these diseases from the point of view often of autoimmune disease, and they clearly are. But then the question is, what causes the immune system to be knocked out of kilter? Okay, and a virus could do that, right? Because viruses can evolve to look like our cells, and immune system combats the virus and suddenly starts attacking our cells. So I think that's very interesting, but it's still, the acceptance is sort of like one step at a time. So now it's sort of Epstein-Barr is a trigger, right? But then the question is, well, why would you just not say it's playing a role in causation? And that leaves open whether it's a trigger that then occurs and then vanishes, or whether it's actually a persistent provocation. And so that's really where we end up, having the next sort of route to progress and understanding these things. And that's something that I think can be resolved fairly easily. You look to see whether the Epstein-Barr is continuing to cause infections in individuals who have, who have the virus, and you might say they're just have that disease like multiple sclerosis because the virus is triggering this altered immune response. So if the virus is still there, still pushing, then the best explanation, I think, or at least the explanation, the tentatively accepted explanation, would be that it's very likely continuing to cause problems, not just a trigger. And this is also important in, let's say, type 1 diabetes, which is another disease that has been accepted as being an autoimmune disease. But in the last 10 years, especially, data have accumulated really indicating, I think, that the certain enteroviruses, it's a group of viruses that include the poliovirus, certain enteroviruses are playing not only a triggering role, but a continual ongoing causal role. And so I think people are now really accepting that those viruses cause at least a triggering role. But if you look at people who've died of type 1 diabetes, and you try to see, are the viruses still there in the pancreas where there'd be damage to those cells that are producing insulin, they are. So that indicates, yeah, they're causing the immune system to sort of cause friendly fire damage, which we see as type 1 diabetes because insulin isn't being produced. But they're continuing to do it. It's not just that they do it, they're hit and they run. They're actually continuing to do it. And that's kind of important, because if they're continuing to do it, then it gives us another option. They're continuing to provoke. Then if you figure out a way of combating them, then there's a chance to actually recover. If it's hit and run, they just cause the immune system to be kind of damaging in a non-functional way. Then if you you don't really have the option to control the pathogen because it may not be there if it was hit and run. But if it's still there, then there may be ways in which you can actually uh, not just treat the symptoms, but actually get at the causes by knocking down that organism, that in this case, the virus, that is continuing to provoke the damage.
That's such a key point because you're saying that these viruses, once they enter our system, they might just be dormant. And even though you may not have active flare ups or major active symptoms, they're persistent and continuing to cause damage in a slower fashion, perhaps, which boils up into type 1 diabetes or atherosclerosis, or Alzheimer's, or what have you.
Right. And, I would say that the language has to be kind of careful here because the language can guide people into one way of thinking versus another. So I think in the case of what you said, it may be dormant. And that is kind of what a lot of times people would think. It's very reasonable. But then you also said, well, it may be flaring up and causing damage. And so we need to understand that, right? And so sometimes the language gets in the way. So, for example, with Epstein-Barr virus, when it wasn't producing viral particles that come out of cells, people were talking, as they do with other herpes viruses, talking about latent infections. But when we actually see what the virus is doing, let's say with Epstein-Barr virus, it's just shifting to a different way of reproducing. It's not latent at all in many of these cells. So instead of producing virus particles that bud off of the cells, it's actually not producing virus particles, but it's causing the cells to divide, and it divides right along with them. And that way it's sort of hidden from the immune system. So it's not latent. It's just in a different reproductive strategy. And so that's really very important to understand because so then it's an active virus that people might think is latent because they're not seeing virus particles, but they label as latent because they're not thinking broadly enough about what viral activity really is. And in this case, viral activity can be shift to cause the cells to divide. You interfere with the cell's ability to control its own replication, to shut down its replication. You interfere with the cell's ability to kill itself, because normally if a cell it finds it's infected, it may say, time to kill myself, right? So I protect the body. Also, the virus causes the cells to not have the sort of protective mechanism of limiting the total number of cell divisions that can occur. And so then these cells essentially become immortal and they're replicating when they otherwise wouldn't, and they can't kill themselves. So in that case, then you've got a lineage of cells that would be infected with Epstein-Barr virus that is growing, even though the body would benefit if it was really kept in a sort of a quiescent state. So if that's the case, then it also becomes a target because it's active. It's a more important target if it's active than in this other uh, way of having activity. And it's also important because if a virus is doing that to the cell is moving towards cancer. Because cancer is a disease in which you have cells that are replicating, they can't kill themselves, and they have the cap on the total number of replications being removed. Okay, so you're already pushing those cells towards cancer. And then if you get mutations, coming back to your first point, then if you get mutations that further abrogate those defenses against cancer, then you can go from a precancerous replicative state all the way past the threshold of cancer. And that's why the viruses that are accepted as causing cancer and many of the viruses that are suspected as causing cancer are very reasonable candidates for bringing into the picture of oncogenesis. And what I mean by that is those, in contrast to the idea that you just have cancer resulting from mutations by chance, disrupting these control mechanisms without causing the cell to just completely malfunction and not be able to replicate. Now you've got a viral infection of those cells that allows those cells to grow to large numbers. They could be expanding that population of infected cells to millions or billions of cells. And now you have the chance in any of those cells to have an additional mutation that pushes the cells closer to and maybe past that threshold for cancer. So in that case, you can really see how the viruses, together with the theory about mutations causing cancer, is much more powerful than just the idea that mutations themselves are the only thing we need to be considering. And so the interesting thing is all of the viruses that are accepted as cancer-causing viruses, oncogenic viruses in humans, they do the same thing, but they've evolved to do it in different ways. Okay, so if we look at cervical cancer, caused by human papillomavirus, and we look at the various, cancers that are associated with the various viruses that are associated with breast cancer, and as well as EBV, then what we see are the same kinds of alteration of those same barriers to the process of oncogenesis occurring. And it's not because the viruses are benefiting from cancer, they're benefiting from persistence, right? If they're causing the cell to divide, they're keeping the cell from committing suicide, and they're removing the cap on the total number of cell divisions, they've got a way of persisting for years, maybe a person's whole lifetime, that they wouldn't have if they were just doing what we refer to as the lytic strategy, which is just reproduce, cause virus particles, and then eventually the immune system recognizes that, rallies itself, and destroys the viruses. In this case, the immune system has difficulty in rooting out those viruses that are more cryptically kind of causing the cells to divide a little bit more, replicating the viral genomes, but not in a way that's attracting so much attention from the immune system. So that really is kind of an important way of illustrating the difference between acute and chronic diseases. The acute diseases, acute infectious diseases, are diseases in which pathogens are replicating, and the race is on for the pathogens to replicate and get transmitted before the immune system shuts them down. That usually the immune system will shut them down in a month or a month and a half. Right. And so in that case, you have acute infectious diseases, things like, common colds and, influenza and so on. And so if the immune system can't get rid of it within a month and a half, then it says the immune system has got a more difficult problem on its hand. And if it can't get rid of a month and a half, the pathogens may have characteristics that essentially allow it not only to last for a month and a half, but maybe months, years. They've kind of got the upper hand in that infection. And so then we're dealing with infectious causes of chronic diseases. So, people have sort of arbitrarily say, what's an acute disease? What's a chronic disease? What's the barrier, barrier to the transition from acute to chronic? And they sort of settle on this about, 1 to 2 months, but there's some vagueness of it, but it makes a lot of sense when you're thinking about the immune system's ability to control or not. If it can control it, usually gets control within a month and a half. If it doesn't get control, if it can't control in a month and a half, then you've got the potential for a chronic disease. It could be lifelong. Okay, and so then the question is, well, why do some pathogens evolve that ability to persist and others don't? And that's a complicated problem. Probably any pathogen would benefit if it could get all the advantages of replication in the short run and then have persistence too. That would be the best of all worlds. There can be some sort of trade-offs that if you're persistent, you may not be as effective in the short run if you're have the characteristics that allow you to last the long run. So that's part of it. It's probably hard to get a balance that allows you to do both. Pathogens that have been in humans for a long period of time, like the tuberculosis bacterium, have got that figured out pretty well. And also a lot of the herpes viruses have got it kind of figured out. They've been in humans probably as long as humans have been humans, and even before then. But the transmission of the pathogens is a factor that could more strongly select for persistence. And in particular, if a pathogen is transmitted by sexual contact or by kissing, sort of intimate kissing, then those pathogens are under especially strong selective pressure to be persistent, because people don't change, sexual partners on a matter of minutes. They change sexual partners on a matter of, weeks, months, years, maybe decades. And so a pathogen that is infecting one individual that's sexually transmitted is really going to benefit if it can still be persistent and transmissible 6 months from now, a year from now, a decade from now. And so when you look at these pathogens that alter these, the function of these mechanisms that are in place to protect against cancer, those pathogens tend to be sexually transmitted or kissing transmitted. Okay, it's not all of them, but there's more of them than you'd expect by chance. And so the idea here is that disproportionately a sexually transmitted pathogen or kissing transmitted pathogen are those are the ones that are going to be found to be more correlated with cancers than your garden variety pathogen in humans. And that holds up. I mean, statistically, in Epstein-Barr's, kissing transmitted pathogen can be transmitted a little bit by sexual, but mostly by kissing. Human papillomavirus, has those same kinds of characteristics. It's transmitted mostly by sex. And, you've got other pathogens like, hepatitis B virus that can be transmitted by sex and also by other routes. So, this is kind of an important issue if you're thinking about educational policies, if you're thinking about how to fund interventions. We can't be ignoring the kissing transmitted pathogens and sexually transmitted pathogens. Education is a pretty inexpensive way to control or at least have some beneficial effect on suppressing transmission of these pathogens. If people know that they could pick up a pathogen from sex or by intimate kissing, it's probably not going to make them more likely to have, those kinds of activities. Probably going to be making them less likely, or if they're going to have sex, maybe more protected sex. And so, this information is actually empowering for individuals at their own level of personal decision making.
That's totally right. And what you're saying is once you get these viruses, they might not be dormant, as I said, they're more
Yes.
latent and then persistently causing damage over the long run, which perhaps just boils up into cancer or one of these
Yeah.
diseases
I
eventually.
mean, you can go pathogen by pathogen. You look at Epstein-Barr virus. Okay, so Epstein-Barr virus now is accepted as a cause of some stomach cancer. It's accepted as a cause of nasopharyngeal cancer. It's accepted as a cause of Hodgkin's lymphoma, at least a substantial portion of Hodgkin's lymphoma. And it's now accepted, I would say, or certainly right at the cusp of acceptance as a cause of multiple sclerosis. It's probably also contributing to lupus and probably also contributing to breast cancer. Not accepted yet, but there's data that really indicate that this is probably the case. And the reasons why it's not accepted. So, for example, with breast cancer, some researchers say, well, we look at the breast tumors and the presence of Epstein-Barr is so slight that it couldn't possibly be causing these cancers, these breast cancers, maybe 1 in 100 cells. How could it possibly cause the cancer? How could it be a major cause? But that, again, is a kind of a source of inconspicuousness or crypticness of infectious causation. Now we know that Epstein-Barr in particular can cause cancers, even if it's only infecting one cell on the tumor. And so this is the story of Hodgkin's lymphoma. Hodgkin's lymphoma, as I was saying, now I think has been accepted as being caused in large part by Epstein-Barr. But if you look at the lymphomas, it's only one cell out of 100 that are infected. And those are the only cells in the lymphoma that are cancerous. So all the other cells in those tumors are non-cancerous. And so that seems crazy, right? Except that if you think about the dynamics of what's going on, you've got a lot of factors that could cause a tumor to be composed of non-cancerous cells, even if the cancerous cells are really instigating the whole thing. So for example, you could have infiltration of those tumors by cells that are moving in. You can have signals cause surrounding cells to replicate. You also have the immune system that may be active in routing out the cells that have signs of infection. So if you have a tumor forming, it might start forming because there are a few cells that cause some other cells to divide, and then the immune system comes in. It may not be that great at it, but it can at least say, I recognize this cell is infected with Epstein-Barr, so I'm going to kill it. and or tell itself to commit suicide, and so that won't be completely effective, but it may be partially effective. So over time, if you get this continual increase in the number of cells that are stimulated to either infiltrate or divide around the cancer cells, eventually you can get to a situation where the tumor is composed almost entirely of non-cancerous cells, which runs against the dogma. The dogma is, oh, well, all the cells in the tumor must be cancerous. But now we understand that you could have only a small portion of cells in a tumor cancer. So you come back to breast cancer and you say, well, people shouldn't be dismissing the idea that breast cancer, that the strong association of Epstein-Barr virus with breast cancer is just some sort of spurious thing where a thing, the tissue is messed up and Epstein-Barr, maybe infiltrating because it's in cells that infiltrate and so on. And so this then should cause people to stop and say, well, let's look hard at those data. And now, even though the studies of infection and breast cancer have not been that well funded in the United States, they've been funded around the world in the last 5 to 10 years. Instead of having just a few studies that indicated that Epstein-Barr was associated, now we've got about 30 studies of the problem, and they vary. A lot of studies don't find a significant association. And some studies find a highly significant association, but we can't ignore whether the ones that don't find a significant association find a weak, non-significant association with Epstein-Barr in general. And that's actually the case. So what you can do is you can look at all of the studies together and you can do, a meta-analysis, which it says, we look at all and we take into account how strong the results are. We see overall, is this virus associated with breast cancer? And the answer is very strongly yes, it is. And that association is something that was generated in spite of the fact that NIH was not funding this very well. It did some funding, but basically there was sort of this tendency not to think this was the way to go. But other countries, because breast cancer is prevalent around the world, other countries were thinking this is something to look at. So we generated these data that lead to this conclusion that Epstein-Barr virus is associated with breast cancer. Some people here I know have said, well, it can't be causing it because it's just not common enough. But then we come back to the idea with Hodgkin's lymphoma. We say, oh no, a virus could be present in only 1 in 100 cells in a tumor and still be causing the tumor. So we can't use that as evidence to dismiss these strong associations that have been generated in the last 10 years between Epstein-Barr virus and breast cancer. And interestingly, the same thing has happened for some of these other viruses like human papillomavirus and this virus that causes breast cancer in mice that gets into humans. If you look at all of the data, although the data are variable, if you look at all of it in one test, in a meta-analysis, you find that these associations are very robust. And so now we just have to go to the next step. We have to say, it's not so easy to come to a consensus on causation just by, looking at characteristics that we think should be associated with causation, like, all of the cells should be infected with the candidate virus or not.
Can you dig into this point a little more when you say for Hodgkin's lymphoma, it could be the case that only 1 in 100
Yeah.
actually have the virus. So what is the virus? Is that cancer cell signaling to the other cells well, they're not becoming cancerous. So what's going on there?
Well, that needs to be clarified, exactly what's going on. There are a number of things that could be occurring. That certainly is one, that's signal. But whatever's, in one sense, you can say, well, we need to resolve this, but we can actually focus on one bit of really hard data, which is, if you look at those Hodgkin's lymphoma cells, the only cells that are actually cancerous are those that are infected with EBV, and the other cells are not infected with EBV, and they're not cancerous. So I think that association has really convinced people that that this virus really is causing the tumor. Now, in answer to your question, there certainly is going to be some infiltration. There's almost certainly going to be stimulation of cells around the cancerous cells that cause them to divide. And then there's also a tricky thing, which is this virus causes, when it infects in this, quote, latent phase, or we should really say lysogenic phase, right, to avoid the term latency. It is functioning in a loop that is outside of the chromosome. Okay. And so that loop may not be transmitted to the progeny. In fact, we know it's not. It's not anywhere close to perfect transmission. So you could have a virus that's infected with Epstein-Barr that was involved in starting cancer. As that cell divides, it could lose the virus. So that may be playing a role as well. But one thing about the Hodgkin's lymphoma is that those cells that are cancerous look different. So if you look at a tumor, it's sort of like, whoa, what are those big cells doing? Oh, those are the damaging cells. And so in that case, it made this the causal role a little more conspicuous. So those cells, you've heard to read Sternberg cells and Hodgkin cells, that conspicuousness made people look at them, and that's what allowed them to realize that those are the cancerous cells. But it doesn't have to be that way. In other tumors, you might not have that clue. It may not be so conspicuous. So you could have tumor cells that maybe look slightly different, but not so obviously different. And unless you really are trying to say, we want to look at every cell somehow and test it to see whether it's cancerous or not and whether it's infected or not, and sort of divide them according to some category that we make up that is based on some kind of difference between the cells. Unless you're doing that, you may miss it entirely. And in fact, some of the people who are studying viruses causation of cancer, I think to try to be sort of conservative to avoid getting people attacking them, they'll have assays that quantify how common the virus is in the tumor. And they'll often say, well, here we have a tumor that is positive for a virus. And but the positivity is low, so it's not causing the cancer. They'll actually repeat that idea. And I think they're being too conservative there. It could be for the reasons I've just said, that those viruses could be causing the cancer, even though they're fairly rare in the tumor. So if there's a problem with being too conservative as well with being sort of too enthusiastic, right? And so one of the problems with saying, well, this virus can't be causing this cancer is that you may lose the option of preventing the cancer or even curing the cancer by really recognizing that they're just some cells that have a target that you would otherwise ignore, an infectious target.
Yeah, and to your point, I think there was a recent study out of Stanford talking about how EBV virus is a leading cause of multiple sclerosis. So it does seem to be becoming more accepted as a theory because that was a pretty
Yeah.
big study done over a long period of time, and
Yeah.
it found that yes, 100% of the people who had multiple sclerosis had EBV virus, but it doesn't mean that if you have EBV, you're
That's
going to
right.
get MS,
Because I think
obviously.
90% of the people who didn't have multiple sclerosis also had the virus. Because,
Right.
I'll get it. We can get it from, kissing, but we can also get it from contaminated materials. And in our society, it looks like as we've, brought Mr. Clean on board to disinfect our environment, we ended getting Epstein-Barr later. And actually with Epstein-Barr, it looks like if you get it later, let's say during adolescence from kissing, it's actually causing a bigger infection. If you get it early, maybe in lower doses, from, glassware or something that's contaminated, or from sort of non-intimate kissing, right? Parent offspring kissing, something like that. Then you get it earlier, in life. And if you get it earlier in life, not clear exactly why, but it looks like you get an immune response that protects you from a lot of the more intense exposures you would have from intimate kissing during adolescence. So it looks like in our society, we have Epstein-Barr causing more severe infections during adolescence than it would occur in some other societies pre-adolescently. And then there are data now showing that if you had multiple sclerosis, if you had infectious mononucleosis, you're more likely to get multiple sclerosis. Okay, so that connection also is there, and it looks like you may be also more likely to get breast cancer, right? So a lot of it, the data need to come from all sorts of um, um, sources. They need to be sort of epidemiological data. They also need to be cultural data. Why does one culture do one thing and one culture does another? They also need to be immunological. They also need to be virological, right? And so, they're just, as well as also in terms of policy. We need to figure out what kinds of interventions could actually reduce the causes of these instead of doing a lot of symptomatic treatment. And, for multiple sclerosis, for example, it's, largely symptomatic treatment, trying to turn down the autoimmune response. And, that's good if it helps individual patients, but it's not as good as preventing at the outset. So we can't lose our focus on understanding the underlying causes of these illnesses, because that's where we're really likely to make the biggest contribution.
On cancer and viruses, um, have you looked at so there's a metabolic theory of cancer, which says that the cancer is caused by mitochondrial dysfunction. Have you looked at whether viruses are attacking the mitochondria and
I've
the
been curious
interplay
about
between
that. I
virus
haven't
and
seen that
mitochondria?
connection. That's a great question. One thing I would say whenever I see a new kind of theory, I would say, well, what causes the mitochondrial dysfunction, which is exactly why you asked the question. And because we need to understand that, right? I mean, if it's not an infection, what is it? And it may be some other factor that could be preventable. I mean, it could be some sort of chemical compound or something that causes the problem. So that's exactly the right question, but I don't think there's an answer yet. But asking the right question is really the way to go, because you ask the question that makes people curious, and then that says, well, we need to understand. We need to make hypotheses about what causes mitochondrial dysfunction, and then one of those hypotheses would be viral effects. And those effects could be very indirect. I mean, it just could alter the sort of reactivity of compounds in the cell, and that reactivity could be causing the mitochondrial damage. So we have to be aware that it might not be direct damage.
Have you looked into glioblastoma at all? And if there's a connection between
Yeah,
viruses and glioblastoma?
yeah, yeah. I mean, that's one that we really need to be bringing as much of a balanced assessment as we can. And the really, Holly Swain, who's working with me on this, has done the lion's share work. She's just sort of told me mostly what she's found, because she's really more the expert on this. But this has a long history, like these other cancers, in terms of trying to figure out whether infections playing a role. This history goes back really 25 years, and early, just after just around the 2000, there were studies that were finding cytomegalovirus being associated with glioblastoma. And cytomegalovirus, interestingly, has not been accepted as a cause of any particular cancer, but it has those same characteristics that I was talking about with Epstein-Barr virus. It's another herpes virus, and it is able to cause the cell to break its cell cycle arrest. It causes it to divide. It inhibits cell suicide, and it also causes the cell to not have the cap on the total number of divisions. So it's got all those characteristics. And so when people were first starting to investigate cytomegalovirus, 25 years ago, there were some early studies that showed association. And then there were follow-up studies, but so often is the case here, that said, we don't find it. And interestingly, then one of the molecular virologists, who really is doing some of the most reliable work, teamed up with the person who found it first and said, well, we're gonna do the most sensitive technique we can do on this. And they did it, and they found it. Okay, so the idea was that it's people who are not, for one reason or another, the assay was looking at the wrong stage of life cycle, weren't using sufficiently sensitive technique. So 10 years later, there was a big meeting where they gathered all the people who were studying this, and they said, can we come to a consensus? And they published that consensus a little over 10 years ago. And the consensus was, it's playing a role. It has these characteristics. The data, the best interpretation is it's playing a role. And so that got some attention. And then other people started coming in and saying, well, I know how to study cytomegalovirus, and I can get some cancer. And they weren't finding it. So the whole thing started over again. So so now we're sort of still kind of stuck, but there are a lot of really interesting studies. And some of these people who are working on this have been doing studies in the Karolinska Institute, and they're looking at antiviral treatment of people who have been diagnosed with glioblastoma, and they're finding prolonging of life with antiviral treatment. There's still some ambiguity because antiviral treatment can also shut down normal replication, right? It shuts down the virus, but also shuts down normal. So one possibility is, well, maybe it's having some effect because it's shutting down the normal replication of the cancer. So there's still some things to work out. But, weirdly, those data are kind of being ignored by a lot of people who feel they're experts on glioblastoma. And, these drugs are really cheap and easy to use. And I would think it really just ought to be something that is part of the normal protocol because it looks like it's having an effect. Glioblastoma has got this very tragically short period of time between diagnosis and death of a few months. And this increases, the data are indicating it doubles or maybe even triples the length of time people can survive, but it's still not fantastic, but it may give people an extra year with this treatment. And, it could also be that the treatment a start of something more effective. So this is a really good, and I'd say a sort of a sad example of how, people were working really well to try to team up and share ideas and brainstorm on this. And they were making progress towards a consensus. And then now it's kind of slipped backwards a little bit. But I think that, people are still studying this intensively, maybe not so much in the United States as in like at the Karolinska Institute, where a lot of some of the US researchers are teaming up with people there. So I think this is going to get solved, but we'd like it to be solved fast, quicker rather than a shorter period of time. But I'm afraid that's sort of the story with most of these cancers and also most of the chronic diseases is that this is just a prolonged period of time, typically decades of between the first study that's attracting attention and a consensus. And, our funding structure doesn't really work that way. Our funding structure sort of works by sort of saying, well, we're going to fund this for, 5 or so years. And if there isn't progress, we're going to sort of shift to something else. And this also occurs even on the longer, broader scale. So, for example, when Richard Nixon proposed the war on cancer, there were people in sort of a virus camp and there were people in the mutation camp, right? They shouldn't be separate camps. People should be looking at both together, as we've been saying. But there were these two camps, and they weren't very nice to each other. Peyton Rouse got a Nobel Prize in the 50 years after he found the first viral cause of cancer in chickens. Okay, and when he got the Nobel Prize, he said, "I hope this puts to rest all those silly arguments about mutations causing cancer because a virus causes cancer." Well, that was not very cooperative and inviting to people on the other side. So basically what happened in the 70s, it was, the greatest advances in understanding cancer arose from people studying oncogenes. And so then the sort of shift happened by the end of the 70s towards it's all about oncogenes that are mutated. And so we get cancers and then other mutations as well. And so then the people studying viruses were sort of saying, well, we don't really think viruses may cause human cancer, but they're an interesting model. They continued. And then just about the time that the people who were sort of in the antivirus camp were winning the battle in the 70s, then you started getting consensus around at least some cancers in their 80s that were accepted as caused by infection. So cervical cancer, for example, was one of these. Burkitt's lymphoma is another example of one of these. And then within another decade, liver cancers and nasopharyngeal carcinoma, and stomach cancers, were accepted as caused by infection. So what happened there is you had this kind of sense that, we might do some funding of viruses, but really we're going to focus on the mutations and the oncogenes. And then that's where their funding shifted to with a very sort of very small amount of funding towards infections. And then that has sort of built up a little bit, but still, I think that the funding will influence the extent to which these slowly or rapidly are able to come to a consensus. These various researchers and people looking from the outside at the various research programs. So that's sort of where we are on some of these things. Now, some of these ended up being accepted fairly quickly because I think human papillomavirus was accepted as a cause of cervical cancer. Then when human papillomavirus was found in oropharyngeal cancer, then it was a much smaller hurdle to get acceptance. So we're really only talking about 5 to 10 years between when it was found, because sort of people think, well, the mucosal system is very similar, let's say around the tonsils and to the, urogenital tract. And so the virus is there. And I think there wasn't the resistance. It wasn't as big of a cancer research topic as, some of these others like breast cancer. So I think that ended up happening quickly. Another one that happened quickly was liver cancer, because in liver cancer we knew there was something that was transmitted in the blood supply that was causing cancer. So people were looking for it. They first found hepatitis B, but even once you account for hepatitis B, then they realized there was still some other risk associated with blood transfusions that was causing cancer. So everybody's looking for this other virus. And then they found hepatitis C by just looking for genetic material that wasn't human in those cells. And when they found that, they said, oh, this is not human and it's a viral gene. So what is it? And they realized, well, it's another hepatitis virus, so we'll call it hepatitis C. So that was very rapid because everybody's looking for it. Another one that was rapid was cancers that are referred to as Kaposi sarcoma. And those were associated in gay men, but not in other AIDS patients, gay men who had AIDS, but not in other AIDS patients. And so people saying it must be something that's transmitted in this group. And so the search was on to find that virus. And so they found a new herpes virus, this Kaposi sarcoma associated virus. So sometimes it goes fast if the sort of the attention says it must be a virus and everybody agrees on that. But then if the attention is focused on other causes and you've got sort of embedded interests, then it can be very prolonged. So I think that's what we're dealing with now. But it's interesting that if you look at all the common human cancers, for every one of them, there are viruses that are associated with them in some studies. Yet we're still sort of accepting 20 to 25, maybe even 30% of cancers as caused by infection, depending on who's accepting or not. And this means that it could be that most of the remaining 70% could be caused by cancers. We just don't know. But if they are caused by cancers, they're probably caused in a much more cryptic way than the virally caused cancers that we accept as virally are caused by viruses. Okay. So that was not a very clean sentence, but hopefully you see what I mean.
So, what I don't understand is that why is the oncogene theory so persistent? Because I think there is evidence to show that even though some people have oncogenes, they don't end up developing cancer. Is that right?
Um, I'm not sure on that, but I
Or
think
some of
on
the, maybe oncogene is not the right
yes,
word, the
I think that's probably,
mutations.
I think that's
Yeah.
right. And it would make sense because even by the oncogene theory, it's not just one oncogene, it's going to be a collection of them, right? Typically, unless the cell is already right at the edge, right? Then it could just be a mutation in one that would be apparent. I think the answer is because it's partly right. I mean, you can show that it is playing a role. And so the real issue is why are people arresting their curiosity about causes of cancer just because we understand what a big chunk of causes of cancer are, with the understanding of the role of oncogenes. So again, it's sort of, I think what we're finding is that what we will find is that a lot of these different camps have part of the story, right? It's just that unless you look at what's missing, you might not be motivated to say, well, we need to fit together another part. It's sort of like if you look at a jigsaw puzzle and you say, oh, well, I've fixed this part of the puzzle, and you're just focusing on that and not looking at the rest of the jigsaw puzzle, which has a lot of empty spaces.
So when everyone talks about breast cancer, you talk about mutations like BRCA1, I think. Have you looked at the the study between these mutations and the viruses that you say are pretty associated with breast cancer?
That's a great question, and that needs to be done. Sometimes there is information on that. I think with the BRCA mutations, they're involved in uh, compromising the repair process. So they're going to be generating mutations. And I haven't seen any information, and I wouldn't necessarily suspect that there's any direct interaction between those mutations and infection. But I think the indirect interaction is the one to be looking at, which is that you're getting more mutations, but the fact that you're getting more mutations and individuals are more vulnerable to cancer doesn't negate the possibility that what is also pushing the system is an infection that is getting you closer to cancer so that you'd get the mutations being higher in frequency and thereby, causing the cancer to occur sooner or occur when it otherwise wouldn't because viruses are pushing the cells towards that limit. So I think that would really need to be looked at. Again, it's sort of like you've got part of the answer, you want to be asking, are there other parts of the answer, even if you've got a strong indication that there's a genetic influence.
That's right. Yeah. So why haven't there been vaccines against some of these sexually transmitted pathogens?
Oh, there are, and that people are looking at it. For example, there's this vaccine against cervical cancer, against papillomavirus. Okay, so that's great. There's a vaccine against hepatitis B, right? And that works pretty well. And there are people who've been trying to get a vaccine against EBV, and it's been a little bit difficult. But I think what's actually one way to look at it is it's going to be more difficult to get a vaccine against a lot of these viruses, even though there's been some success, because these viruses have evolved to persist in the face of the immune system, and the vaccines act by stimulating the immune system. Okay, so these viruses are going to be less able to be controlled by vaccines than would viruses that are causing acute diseases we want to prevent, right? So if you're thinking about these acute diseases like influenza, the immune system controls it. So if you just give people a vaccine, and if you get the right makeup of the vaccine, which is tricky, then you get an immune response that is able to control the virus much as the immune system would in response to the virus, right? So I think that we're used to vaccines being effective for acute infectious diseases. And then now as we're looking at vaccines that are geared towards controlling chronic infectious diseases, they're just going to be more difficult. They're probably not going to be quite as effective just because the viruses have evolved to be less controlled by the immune system.
Okay, so then
Doesn't mean we should stop because we have
right.
had successes, right?
So then practically speaking, say I go and get tested and figure out whether I have some of these viruses and bacteria or not, because you can get screened for them, you can get tested for it. And if I do find that I do have some of them, they're probably latent and don't have active symptoms, what is one supposed to do about it?
Well, I think it has to be dealt with on a pathogen by pathogen basis, right? So for, let's say, varicella zoster virus, which is this herpes virus that causes chickenpox, but also shingles later in life. Now we've got a vaccine that is effective in protecting against shingles, even though you're infected with varicella zoster. In this case, you don't really need to be tested for varicella zoster because almost everybody's got chickenpox, but not everybody. So so generally, basically, you just give the shingles vaccine when a person gets to an age where they're more likely to be having a resurgence or recurrence of latent chickenpox virus. So that's a good example of one in which, yeah, you, especially if you've had chickenpox, and I would say if you had chickenpox later in life, it's probably going to be more severe, and it'd be a good idea to be especially raising the priority for getting the shingles vaccine. And so I think that it's just not, I don't know of data that really give you a clear-cut answer, but I think that the answer to your question is going to be that it will become clearer for each particular infection when we have the vaccines, we understand how they work, to uh, indicate or to advocate for the use of a vaccine. I mean, with SARS-CoV-2 vaccines, now it's pretty clear that you get the additional vaccines, you're going to get some protection. But like with influenza, the protection is not necessarily that you're going to be free from infection or free from disease, but if the disease occurs, it would be milder. And with SARS-CoV-2, the issue now is to what extent are the viruses actually persisting or not? And, when we're trying to understand what's causing long COVID, one of the possibilities you've got these persistent viruses. So, but the data are indicating that, the additional vaccines are protective. It's just that they're not perfectly protective. So that then may cause some people to decide not to, get additional vaccines and, additional doses, booster doses of the vaccines. And other people, may have different mix of concerns or fears that would be leading them to a different conclusion. But I think that the general answer to the question is just people need to be clearly thinking about what the evidence is for each particular vaccine and each particular disease, and very likely the recommendation will be specific for not only disease but also for individuals. If a person is immunocompromised, for example, then the risk of shingles is much greater than if they're not, right? So if a person is likely to be immunocompromised, it may be good to have a shingles vaccine earlier rather than later.
What about EBV or chlamydia, pneumonia? Like you mentioned EBV
Don't
multiple
have
times. Like,
vaccines
what do
for
you?
them. Yeah, when we get vaccines
So,
for them,
what
I
is
mean,
one supposed to do in the meantime?
yes, okay. So with chlamydia pneumonia, for example, it's a respiratory tract pathogen. If you are just a little more conscientious than you might have been previously about paying attention to respiratory tract pathogens, making sure you treat yourself better, getting enough sleep, and maybe taking antibiotics when you otherwise wouldn't, you're likely to reduce the exposure to chlamydia pneumoniae. We talked a little bit about the periodontal pathogens. That's a great one in which if you actually do take actions to reduce the periodontal disease, then you're likely to be getting lowering your risk of these chronic consequences. Okay, we talked about flossing, for example, as being something that would be a way of protecting against the chronic diseases, which are obviously very serious. If you're thinking about Alzheimer's and thinking about diabetes, thinking about cardiovascular disease. So I think it's going to be situation specific. No vaccines available for chlamydia pneumonia yet. And although people have been trying to get a vaccine for EBV, there's not a vaccine yet. But I think when the vaccine's available, then the tests will be done and we'll see whether or not the EBV viral load is lower if you're having a vaccine, or if you're protected against new infections. It'd be the same kind of thing that has been done on SARS-CoV-2 vaccines to try to figure out how good is the protection. And then those data will influence recommendations. Of course, it all depends on people being, who are responsible for making those recommendations, having a balanced view of vaccination, right? And so, vaccines do have potential negative side effects, but the question is, what are the benefits versus what are those side effects? And the vaccines in general have been very safe. The safety has been, leading, generating the possibility of serious side effects down in like the 1 in 100,000, 1 in a million, 1 in 10 million category. And if you don't have vaccinations, the risk of really serious disease is way, way higher. So I think people need to have a balanced view about that. And I'm not sure where we're going to
And
be in
I,
this
s-
country here in the next few years.
I suppose one other thing people could consider is that if they do end up getting unfortunately one of these chronic diseases and they do have the virus, you could treat it with some of the antiviral treatments, which
Yes,
might
right.
prolong. Yeah.
That's right. That's right. And so, that sort of brings back the, glioblastoma story. And also, yes, if you've got a severe problem with a persistent infection, or even with the first infection, if you can get it early enough, yeah, you can take these antivirals for, SARS-CoV-2 and for influenza that are going to be reducing the chance that the virus is going to be causing problems.
Do you think this is one of the reasons people have cancer relapse? Because the virus is probably still in the body?
Yeah, well, if the virus is causing the cancer, yeah, that would certainly be very reasonable explanation, right? Um, it could be that once the cancer develops, you lose the viruses. Once the cancer develops, you got all these cells, you could have additional mutations, and you could lose the virus. And so in that case, it wouldn't have that effect. But certainly, I think in those circumstances, it's likely going to be a combination of things. If you still have the virus there, it's still pushing those cells towards the cancer threshold. Even after you pass the cancer threshold, you still have that influence. But once you pass the cancer threshold, or even approaching the cancer threshold, you have all these cells. So you could have mutations that are going to accelerate the progress, or the advancement of the cancer, but also mutations that might make the cells less be able to reproduce with less exposure to the immune system. So if you've got a virus, let's say in a cell, cancer cell, it's still going to be imposing a load on the cancer cell. And it also is going to have some increased exposure to the immune system. So you could imagine, although I don't know data that really show this, you could imagine that the viruses may tend to be lost. And then any resurgence would be just a result of, the let's say therapy, knocking down the cancer to a small number of cells, but those cells would be resistant to therapy. And then those cells could bounce back. Maybe they would bounce back slowly, maybe they bounce back quickly. If those cells were more compromised in terms of replication, they might bounce back more slowly, even without a virus. So I think the jury's open on that. I just don't think we know. But it's very reasonable to expect the viruses could be playing a role, but it's also possible that it's going to be more a question of mutations now that are have upset things, led to those cells being rapidly replicating, but now those cells being a large population, even after the control by, let's say, chemotherapeutic treatment, you still have a lot of cells, even though you don't have necessarily billions of cells, you might have hundreds of thousands or millions of cells. So that those essentially could be ticking time bombs for additional mutations that would then lead to the resurgence. So I think it's just going to be specific to individuals as well as the cancers. And, this sort of comes back to individualized therapy to look and see what an individual's circumstances are, right?
All right, Dr. Ewald, any last practical tips for people in terms of how to prevent some of these
Well,
viruses and pathogens
yeah, I
or
guess my last tip would be that we have had an immune system that has evolved over hundreds of millions of years. And we've inherited this from all of our ancestors. And it's an amazing system. And it just may be that if we could keep from compromising our bodies and letting our immune system function well, we might actually avoid a lot of these things because it just may be when we push ourselves, we stress ourselves, there are a lot of things that our immune system might be able to control that couldn't. We reduce our exposure to pathogens, reduce our stress, and thereby reduce the compromising of the immune system that occurs during stress. And the other thing we haven't talked much about is diet. So diet ends up, it's very clear now, it alters our microbiome. And if the diet is favoring organisms that are mutualistic, one of the things those organisms do is to protect against the pathogens. Okay, so if we can merge diet, and we haven't talked about exercise either, but exercise is something a lot of experimental studies have been done on that show that if you exercise, you get enough sleep, and you have a good diet, you really can be very resistant to a lot of these processes. So and that resistance may really involve preventing the problems from occurring. So I would sort of end with that, especially since we haven't talked that much about that, but as being part of the sort of integrated picture that we need to bring to understanding health and disease.
Stay healthy, folks. I mean, that's
That's
always
right.
constant.
Yeah, that's
Be
right.
healthy.
Yeah.
Last question. What directions of research are you excited about in this area, or any other things that we haven't covered that you want to touch on?
Wow, I every time I look at literature, I'm excited by what people are doing that may not have been recognized. So I would say that we're getting close to this point. And actually, we've always been working towards, but we've always been a little bit behind the curve. But we're maybe staying up with the curve a little bit more on this idea that we need to really integrate our understanding from all sorts of research areas. And I think people are open to this, partly out of frustration that we haven't solved some of these problems. And the frustration is not bad. It's a good side of frustration that makes people say, well, let's think more broadly about this. So I think that's what I'm really encouraged about, is this sort of openness to a more broad perspective rather than the narrow perspective, I have the answer and you don't, that kind of thing.
Yeah, I totally agree. I think we need more unified theories and more pieces of the puzzle that can fit
Yeah,
together and hopefully form the entire puzzle.
great.
Well, any other last thoughts?
I think that's it for me. Other than
Great.
it's been great talking with you.
Great. Thank you so much. This was wonderful.
Hi, everyone. If you wish to be notified of future podcast episodes and my writing on longevity and the frontiers of biology, please subscribe at livelongerworld.com. And if you're enjoying the show, please leave a rating on Apple, Spotify, or elsewhere. Thank you for listening, and I will see you next time.